Hi everyone, welcome to our review class on gene expression. And sorry, I'm still recovering from a really bad cold, so my voice might be crappy, I might be coughing from time to time in this video, and I apologize for that.
Alright, now let's take a look at what we have here. You should be able to identify that these are a whole bunch of eukaryotic cells, and that is because we can identify the nucleus within the cell. So nucleus, it has a nuclear membrane or a nuclear envelope, which is a phospholipid bilayer. And within the nucleus, we have our genetic materials, which is DNA. So in this case, the DNA is shaped in a state called chromatin. It is not the most condensed form of DNA, as the most condensed one is called chromosome, and it only shows up when the cell is undergoing division. But right now we're looking at transcription and translation, so there's no need to make chromosome. And within the cell, we have 23 pairs of DNA molecule, so that here we should have 23 pairs of chromatins. And let's zoom in onto one chromatin. So this should be a DNA molecule. And remember, the shape of a DNA molecule is a double helix. So let's review the shape. First, we're going to look at the process of transcription. So we need the help from an enzyme called RNA polymerase. It has two functions. It will bind to the promoter site on the DNA, and then it's going to open up the DNA double helix. So that is the first function of RNA polymerase. But it's also going to bring in new nucleotide through base pairing. So the first base here we're interested in is a T, and it's going to bring A base, so that is a nucleotide. And here what we're making is ribonucleic acid, and what we had as template is deoxyribonucleic acid. So the structure on the sugar, which is not shown here, is also a bit different. For the DNA nucleotide on the sugar, we only have one hydroxyl group, but then for the sugar on the RNA here, on the sugar, we should have two hydroxyl group. Alright, so now the RNA polymerase is going to bring in one more nucleotide through base pairing. So A is going to be paired with U, that is a ribonucleotide. And then we are going to connect the backbone as well. So this is all done by our RNA polymerase enzyme. And next we have a G base paired with a C through 3 hydrogen bonds. So I'm going to go through all the base pairing. And let's move on to the next. Alright, so we're going to stop here because the RNA polymerase is almost at a terminator region. So one more nucleotide, and that's it for transcription. However, what we get at this point is a pre-mRNA. So this is not a mature mRNA because we still need to do modification on it. And we need two types of modification. The first one, or the first action, is to add a 5' to the mRNA molecule. And we also need a 3-poly-A tail added on to our mRNA molecule, so that this molecule will not be destroyed by the enzyme in the cell, and the molecule knows where to go, how to leave the nucleus and go to the cytoplasm. So once the modification is done, we have mature mRNA, and then it's going to leave the nucleus. So this is our mRNA, and it's undergoing modification. Now it has left the nucleus. And then we can proceed with translation, which is the process of turning the genetic information on the mRNA into actual protein. So to start the translation, we will need the help of an enzyme called initiation factor, which is not shown here. It's going to recruit one small subunit of the ribosome. So here, this is our small subunit, and this is our large subunit. And another RNA called tRNA, which is transfer RNA, is going to bring in the first amino acid into our ribosome. So this is actually not the most accurate animation. What really happens is what we talked about in class. The initiation factor is going to help this small subunit bind to the 5 cap, and we will have our tRNA bringing in our first amino acid, which is methionine here. And then lastly, the large subunit will bind into the same region. Alright, so now let's go back to our ribosome. Inside the ribosome, we have 3 regions. We have an E for exit, and then P for polypeptide transferring region, and then A for the attachment of new tRNA. Alright, so right now we have our first amino acid in the P region, and this is our codon here. So AUG is the start codon. And that is the case all the time for all the cells in all types of animals. And we have anticodon, which is the complementary codon for AUG. So tRNA is going to match with the start codon through the anticodons, and it's going to bring in the amino acid here into P site. And this is the only time where the incoming tRNA will enter the P site. For the rest tRNAs, the new ones, they will actually enter the A site. And once the new tRNA is here, it's also going to match the codon. So CCA is the codon that codes for the amino acid proline here. And once the new tRNA is here, our P site is going to transfer the amino acid into the new amino acid through peptide bonding. So now we have a dipeptide, methionine and proline here. And next, the ribosome is going to move, and then we are going to release the tRNA, which is no longer useful. In the meantime, we have a new tRNA bringing in a new amino acid. So glycine is coded by GGC, our codon here. And the same, in the P site, our dipeptide will be transferred into this new amino acid, and now we have a tripeptide. The ribosome is going to move again, release the tRNA that is no longer useful, and then transfer the peptide into the new amino acid on the A side. Alright, so ribosome moving again, and then we are transferring the peptide again. So all of those are facilitated by the enzyme that is called the elongation factor. Finally, now let's look at UAG, which is the stop codon. So it's not shown here in the animation, but the ribosome will also move to the stop codon. And then we will have the enzyme release factor coming in place, which will disassociate this peptide from the tRNA, and that's what's happening here. The tRNA will also disappear, and the 2 subunits of the ribosome will disassociate from our mRNA. And that's the end of translation. Now we have a protein which will be folded into the shape that is useful, and then this is a functional protein.